Hello, and thank you for attending this session entitled RDCA-DAP Facilitating Collaboration in the Pre-Competitive Space, Case Study in Friedreich's Ataxia. My name is Megan Cala, and I'm a postdoc of Pharmacometrics on the quantitative medicine team here at c-path. And I'll now turn it over to my co-presenter, Christian Rummey, for his introduction.
Thank you, Megan. Christian Rummey, I'm a scientist and statistician, and my story with FA started more than a decade ago when I was involved in one of the first phase 3 programs that were run in the disease, a compound called idebenone. And then really I have never left the space ever since, and for the last couple of years been a statistician on one of the two large natural history studies that are available for Friedreich's ataxia. And the work I've been doing there is of course funded by FERA. And we're going to do this a little bit back and forth, so Megan will introduce you to the platform and what we're planning to do with it. And I will speak about the disease and the endpoints and how to measure really FA. So what is FA? It's a life-shortening degenerative neuromuscular disorder. It affects about 1 in 40,000 people, and it's easier to remember if you say there's about 5,000 patients in the US and about 10,000 supposed to be in Europe. It's the most common hereditary ataxia. Its inheritance is autosomal recessive. So carriers are usually unaffected or asymptomatic, and children have a chance of 25% to inherit the disease. The genetic cause is a repeat expansion in the frataxin gene. And this expansion usually has lengths up to 30, and that's the normal length. But in FA patients, it goes up to more than 1,000, sometimes 1,700 repeats. And these repeats cause a frataxin protein deficiency, leading, among other things, to iron accumulation in the cells, which then leads to cell death. And it's important to notice that among the neurological endpoints that we'll mainly discuss today, FA patients also suffer from cardiomyopathy, which is the leading cause of death. It's just that the development of endpoints in the cardiac phenotype has turned out to be much more complicated and is not as advanced as on the neurologic phenotype. And since it's a multi-system disorder, it affects also other symptoms, leading to an increased number of diabetes. So this genetic defect, the number of GA repeats, you can correlate with the age of onset of a patient. So on the y-axis, you can see more repeats lead to earlier age of onset on the x-axis. And this correlation works pretty well if you look at this whole natural history scale. However, it's slightly modified towards earlier age of onset. And if you look at the red group, then you see that the correlation doesn't really work anymore in really young patients. So the disease progression in FA starts with diagnosis, and that at diagnosis, the patient is usually asked, when did you perceive your first symptoms? And that happens typically about 2 to 4 years before diagnosis. And then after diagnosis comes a period of loss of walking capacity that then leads to use of walking aids and eventually loss of ambulation. And after that, people become full-time wheelchair users. Now it's important to note that many of the measures that are used today focus on walking and balance and gait, but about 50% of the overall population of people living with FA is actually non-ambulatory. So it's very important to also develop measures on this side. Now I've mentioned age of onset. I have dashed lines here because age of onset can really occur at any time in life. It happens in really young children and as you've seen in the previous slide, also during adulthood. And I've tried to visualize that here by showing age of onset as a circle and age of diagnosis as a blue dot. So there's really early patients, and I've shown here up to 30, but in the database we have people with onset up to 70 years of age. And you can see easily if I put the first milestone, loss of ambulation, on the scale, then you see that this can happen very early, in basically early teenagers in the most severely affected, while people who have an adult onset basically can walk for decades after onset. Now, when you think about the measures and the development of the disease, it's clear that the younger affected patients are at their onset, the more this will affect with their neuronal development. And there are several thresholds that have emerged that are somewhat separating the clinical presentation of these patients. And certain categorizations have been proposed, like everyone who has an onset earlier than 8 years is early onset, and then we have typical, intermediate, and late onset. So there's a huge diversity. That's the point I'm trying to make here. If you look at the distribution, most patients are actually diagnosed between 8 and 14 years. But of course, there is a huge selection bias in this graph, as of course, the milder patients are, the less motivations they have to actually participate in such a study. Now, the next slide, I want to talk about the clinical trials that have been collected in the database at c-path. So we have the early studies on idebenone that I mentioned before, and then we have 4 other clinical trials. And because the disease is so diverse and patients can behave so differently at different ages, it's important to contextualize and have the background of these studies in mind when you analyze them. And yeah, so that was the introduction into the data and the disease. And now I'm going to hand it over to Megan to speak about the platform that c-path has built around it.
Thank you, Christian. So yes, the FAICD is hosted on RDCA-DAP, and it can be accessed by logging into FAIR Data Services. And you can select the FA aggregated data to get some more information about its contents, as shown in this screenshot here. And this will include some relevant descriptions related to the catalog info, keywords, data dictionary, as well as field level metadata. And also from this page, if data access request workflows are enabled, users can request access to this data set, and it will be delivered to your workspace. The next slide, please. And so to give you an idea on the size and contents of this data set, as Christian discussed, there are 6 clinical trials and 1 long-term natural history study, which amounts to approximately 1,500 subjects and close to 5,500 observations. So this is a very rich database. And the 2 plots on the bottom left show a breakdown of subjects per study, as well as the average number of observations per subject by study. And then to the right, we've summarized the categorical and continuous covariates of interest. These include race, ethnicity, sex, and baseline ambulation status. And for continuous covariates, we have age, baseline age, age of onset, and the 2 GAA repeat lengths that Christian discussed. And these are all candidate covariates that we can incorporate into our analysis as we try to understand the variability in the baseline severity and the progression rate of FA. Next slide, please. And to further interrogate the FAICD, I want to highlight the work of Kurt Michaels. He's a member of c-path's data science team, and he has developed an R-Shiny GUI that's available as one of the RDCA-DAP's mini apps. And this FA interrogator can be accessed under the mini apps tab and launched directly from the workspace. And within this app, you can perform all sorts of data exploration and data visualizations and statistical summaries in a very simple and user-friendly manner. And in this screenshot here, you can see a histogram and a normality check of one of the available endpoints from this data set. And all of this can be executed without having to type a single line of code. And within this app, you can also filter through particular dataset domains to generate subsets for analysis. And this was one of the RDCA-DAP's pilot projects, and it was covered in much greater detail during one of the webinars that was hosted earlier this year and can be accessed on c-path's YouTube channel. And additional analyses can also be executed directly within the workspaces environment using available open source software. So there are mini apps available for both RStudio and Jupyter Notebook so that users can utilize both R and Python respectively via these mini apps. And here I'm showing you an example analysis that I ran of the baseline age across the available studies using the RStudio mini app. And what's really convenient about working within the platform workspaces is that you can easily share scripts and figures and files instantaneously with your collaborators. And if we take a closer look here at these baseline age distributions, we can see that the median age is slightly variable from study to study, ranging from the low teens to late 20s. And as you can see, the range of these included ages also varies from study to study. And this is due to the different enrollment criteria that correspond to each of these studies. And this is important to note because what this also means is the underlying disease severity of these included subjects is also going to vary. And in addition to age, we can also examine the ambulation status of individuals at baseline. And as Christian discussed earlier, the loss of ambulation is an important milestone in the progression of FA and can therefore indicate the diversity in terms of disease severity among patients at baseline. And this figure shows a histogram of the baseline ages of all patients across the entire FA-ICD. And by stratifying by ambulation status, where blue indicates the ambulatory individuals and orange identifies the non-ambulatory, we can see that at baseline, there are many patients that have already reached that non-ambulatory milestone. And if we compare the area, the blue area versus the orange area, as you move from left to right, we can see that the proportion of ambulatory individuals decreases with increasing age. And in this next visualization, we've separated the ambulatory individuals on the top from the non-ambulatory individuals on the bottom. And this figure includes all observations, so not just the baseline visits. And now the colors indicate the 4 different bins for the age of onset. And what you'll notice is that the ambulatory section, there is very little red and blue past the age of about 30. And that's because those earlier onset groups typically progress faster. So loss of ambulation occurs sooner in those individuals. And overall, from this graphic, we can identify the underlying variability with respect to disease severity in these various age of onset groups or age-based populations, which is often the case in clinical trials. And now back to Christian to discuss the various endpoints in FA.
Yeah, so Friedreich's ataxia is usually measured and assessed with neurological exam scales, rating scales. And one of the 2 most important ones is the mFARS, the Modified Friedreich's Ataxia Rating Scale. It's a multi-component scale that covers bulbar function and then upper and lower limbs, as well as balance and walking functions. That's the upright stability score, and which is arguably the most important one, important scale in ambulatory people. Then in the gray box in the middle, you have a functional disability staging that assesses the disease on a broader scale and focuses on milestones. And we have an activities of daily living score, which is a patient-reported outcome. And lastly, we have also direct functional measures in the database. These are mainly assessing walking and upper limb function, and the two most important ones are the time 25 foot walk and the 9-hole peg test. It's important to know that there's quite a few additional measures in development. Just these are the two that we have vast amounts of data of. Now, if you put these measures in the context of my disease progression slide here, you can see that the FARS was actually designed to cover the whole disease range. But if you look down at the subscores, you will see that, of course, certain scales only work in certain disease phases, and balance and walking only works in ambulatory people. And we could show that, for example, upper limb function really is only, or is mostly lost only in the non-ambulatory phase of the disease. And the same is true for direct functional measures, where we have the time 25 foot walk, obviously in ambulatory people, and the 9-hole pegboard test, which is actually the only outcome that seems to work throughout the whole range at the same quality. And with a PRO, it's a little bit the same like with the rating scales, because they do work over the whole range, but they behave differently when people start to bother about different walking functions. And yeah, that's it. So now I'm going to turn it back to Megan to show you the amount of data we have in the database.
Thanks, Christian. And so as for the endpoints available within the FAICD, this figure summarizes the number of unique subjects contributed by the various studies that are available at baseline for each of the endpoints. And all of the endpoints that Christian just discussed are included along the x-axis. So as you can see, the studies vary slightly with respect to which endpoints are included. And the last two bars on the right show combinations of endpoints. So if we were to consider multiple endpoints in our analysis, understanding how that may alter the number of patients and data points that we have to develop our models from. And on the next slide, for the endpoints that are composite scores, such as mFARS, which I'm showing on this slide, we can break those scores down to their item levels. So in the case of mFARS, there are 18 items. So those are the 18 bars shown here. And for each item, we can note the distribution of scores within the population. And you can read this figure like a heat map, where the darker regions correspond to higher scores or higher severity. And what this figure is conveying is the severity of the population at baseline per item. So for example, the lower limb subscore C, and that's the third subscore from the left, the population in this data set spans the severity spectrum. So you have individuals that fall into each of these 5 shades of orange. And compare that to some of the bars in the upright stability or subscore E items on the far right, where some items, the majority of the population is already at the max severity at baseline. And this is particularly important when we're looking at these scales longitudinally, because subjects that have reached the max at baseline can artificially show slower progression or no progression at all. And next slide, please. So what we've discussed today here is really an overview of the types of analyses and of these clinical trial data sets and natural history study that are very important and align our thinking in the development of disease progression models. And these are mathematical models that describe and quantify disease endpoints and use available information in the data sets to identify sources of variability in the patient population. And this is really the current status of this ongoing effort. And as we continue on with next steps, we can also add on a placebo effect model if we can confirm that the data will support this additional model component. And then we can simulate asymptomatic drug effect and use the models to estimate the sample sizes that would be required to detect a significant effect. And lastly, on the final slide, I will end here with the big picture of this effort, which is a clinical trial simulation tool that we aim to develop to be hosted on the RDCA-DAP platform. And with the intended use for this tool being to inform future clinical trials in FA by means of calculating sample sizes, optimizing trial durations and treatment effect times, and answering trial design and analysis questions to support the greater FA community. And with that, we would like to thank everyone for your time here today and attendance, and use the remaining time to answer any questions that have come in. Thank you.